Ctenophora, otherwise known as comb jellies, are mostly found swimming or lurking along the bottom of the ocean. Most of these species live on other animals, for instance, echinoderms, sponges, or benthic cnidarians. Some defining characteristics of this group are they have tentacles that bear coloblasts to capture prey. They are planktonic, unpigmented, and transparent. Also, they are named for their 8 symmetrical tracts, otherwise known as comb rows, made up of fused ciliary plates on the body surface. They are iridescent and bioluminescent. Bioluminescence is the production and emission of light by a living organism, widely found in marine vertebrates and invertebrates. Ctenophora are biradial. They contain 2 tissues. They do not have cephalization. They lack a true body cavity, making them coelomates. They have 1 opening for digestion. They use diffusion for respiration. They are mostly made up of water, which makes a hydrostatic skeleton. They are not segmented and use tentacles as appendages.
Ctenophora are fairly simple animals. Many live only in marine waters and can be found in most marine habitats, from polar to tropical, inshore to offshore, and from near the surface to the very deep ocean. Ctenophora are hermaphrodites, meaning they are capable of self-fertilization. Their gonads are located in the parts of the internal canal network under the comb rows, and eggs and sperm are released via pores in the epidermis. Fertilization is external in most species. Ctenophora are carnivorous and prey on small zooplankton, small crustaceans such as copepods, amphipods, and even euphausiids, and larvae of other marine invertebrates such as clams and snails, using a transparent stomach covered by specialized microscopic sticky structures known as coloblasts that adhere to the prey.
The size of ctenophoras range from 17 to 20 millimeters in length. It has a pear-shaped body about 5 to 20 millimeters in diameter, broader at the aboral end and narrower at the oral end. Ctenophora are the largest animals that use cilia, which are hair-like structures, for locomotion. The cilia are fused into comb rows. The comb rows themselves have multiple functions. Some ctenophores use these comb rows for feeding and swimming away from predators. Statocyst, an organ that dictates orientation and balance in organisms, also plays an important role in the activity of the comb plates. The strokes of the cilia on each comb plate beats toward the aboral surface of the animal. The ctenophore swims with its mouth forward. There isn't enough study done to determine whether or not ctenophora are either protostomes or deuterostomes. The development of the fertilized eggs are direct. In other words, there is no distinctive larval form, and juveniles of all groups generally resemble miniature cetipid adults.
There are estimated to be between 100 and 150 species in the phylum Ctenophora. There are 2 classes in the phylum, Nuda and Tentaculata. The Nuda class consists of only 1 order, Boroida. Boroida species lack tentacles throughout their entire lives. The Tentaculata includes 8 orders, Cestida, Cambogeta, Cryptolobiforida, Cetipeda, Ganashida, Lobata, Platytinida, and Thalassocalicida.
The central nervous system for all ctenophoras consists of a nerve net.
Cetipeda species are thought to be the most primitive. They retain their tentacles throughout life. The order Platytinida is a benthic group that lost the tens or comb plates as an adaptation to the benthic lifestyle of the adult. The orders Cambogeta, Cryptolobiforida, Thalassocalicida, and Ganashida are monogeneric and were proposed for forms that did not meet the definitions of other orders. The order Lobata can be easily recognized by the presence of large oral lobes and auricles and by the absence of tentacular sheaths. The order Cestida has members with a ribbon-like body and modified tentacles that run along its length.
These are our 2 phylogenetic trees. The first one compares the relationship of these organisms in terms of genes, specifically 16S rRNA. The second one compares the relationship of different organisms in terms of morphological observation. Our research, as well as our trees, show a discrepancy between the relationship of Ctenophora, Cnidaria, and Porifera. Our primary article by Wahlberg, Thondison, Ferris, and John Delius described 3 main hypotheses shown in the trees above. In hypothesis A, Ctenophora is in a sister group with Cnidaria. In hypothesis B, Ctenophora is in a sister group with Bilateria, a group of phyla that we did not delineate, with Cnidaria branching off earlier. In hypothesis C, Cnidaria and Bilateria group are sisters, while Ctenophora is branched off earlier. Hypothesis A fits the best with our 16S tree shown below it, because Cnidaria and Ctenophora are sister taxa with Porifera as the most primitive outlying group. Hypothesis B fits the best with our character tree shown below it. Although Bilateria is not shown, Ctenophora is not a sister with and occurs after Cnidaria. Porifera is again the most primitive outlying group. Finally, the C hypothesis fits with the phylogenetic analysis of the articles shown below, suggesting a sister group relationship between Cnidaria and Bilateria, with Ctenophora branching off earlier. There are various reasons for these discrepancies. Synapomorphies can often be mistaken for convergent evolution. Also, RNA, DNA, and morphological observation can differ from one another, creating different phylogenetic hypotheses.